Good evening, participants. Am I audible to you? Yes? Yeah, so just interact I don't know whether the internet streaming is good or not, so only I'm interacting like this. Good, thank you for your response. So can we go for the last session topics? So before going to the last session topics, I can tell you what exactly we'll be discussing it on today. Today we'll be discussing on day 5 on sequence analysis, especially sequence alignment and its types. And we also see PAM and Blossom. Okay. So these are the concepts we'll be discussing it on today. So can we go to the last session topics? And participants, I just apologize. Like actually, whatever videos provided or just viewable. Okay, so just up to the course, you can learn and for appearing the test. Okay, like if you have missed any sessions, the coordinator is sharing you the video. Okay, so actually you have to attend on all the days. If not possible, so whatever recorded videos you can utilize it. I suggest you to take notes. Okay, so watch the videos and take a note and whatever you want to prepare, or even if you have a PowerPoint, you can prepare a PowerPoint, okay, simultaneously so that you won't forget. So this is the way we can register in our mind. Okay, so just saving the recorded videos or even whatever videos in the YouTube won't give you a permanent knowledge. So I suggest you to prepare PPTs or lecture notes or whatever your handwritten notes to collect the concepts. Okay, so I realized. Okay, then tell me, participants. I think one is has unmuted. Anything you want to discuss with me before starting the session? Laiba Osmani, do you have any doubts? Kindly mute. If you don't have any doubts, kindly mute. Okay, thank you for your response. So, can I go for discussion of last session topics? And do you have any doubts? We have completed 4 days sessions, isn't it? We have completed biological databases completely. Now we are going for the sequence alignment, analysis and alignment. So if there are no doubts, we can discuss the last session topics. So what exactly did we cover in the last session? Cancer gene expression database. Very good. Fine. I got answer. And how about the others? What else? Just cancer gene expression database. Then I have to ask you, what is the name of the cancer gene expression database? We have seen so many databases also, isn't it? Secondary databases, types of secondary databases we have discussed, and protein-protein interaction databases. Good. So you are telling overall classifications, isn't it? But also remember the examples. Okay, can be a cancer gene expression, secondary database, protein-protein interaction, string. Very good. So for protein-protein interaction, we have seen string database. Can someone tell me for secondary databases? Second example for secondary databases. HDP. Okay, no, this is for still comes under uh, drug protein interaction databases. SCOP and CAT is excellent. Okay, so these are examples of secondary databases. Good. So can we go for example for cancer databases or cancer gene expression? However you take. Good. We also seen so many tools. I think Kirtana K has accumulated so much information with her. Even others can remember all this. Okay, so mentioned Open Babel, Babelbin. We have seen cancer databases like My Cancer Genome. Okay, we have also gone for I Square ITED server from Raghava. Okay, Professor Raghava has developed multiple tools and databases. Even I am working under the guidance of Professor Michael Komiha. He also has developed nearly more than 25 databases and 25 tools. Okay, so it's a very largest collection from any of the Indian professors. We also seen about C Bio portal, online Mendelian inheritance in man database, GEO profile, gene cards, TCPA, GPA server. I think Kirtana is following my principles. Even when I attend a online or offline workshops, I take everything in Microsoft Word, or I just copy, even if it is possible, even I can copy a slides and put in my PPT. Okay, but if you put like that, if something is mine, you can show the source or of, okay, that will be better for you. So most of the topics, whatever I take is self-prepared. If I show something like textbooks, you can show the source of the textbooks. Okay, then it will be safe for you. If you like to deliver it, if you want to learn, okay. If you want to deliver any talks, you do like that. Okay. Then secondary databases like SCOP, CAT, okay, protein databases like String, Stitch, excellent. Okay, so I think we got all the last session topics. That now can I go ahead on today's session? So today's session is something new, isn't it? So I just show you the schedule, what exactly we for the subsequent classes. I think this is the last session for this week, isn't it? So we have a Saturday and Sunday break. So the Saturday Sunday break are majorly meant for recapping all the session contents. If you have missed any contents, I mean, if you have missed any classes or any content even in a specific class, okay, you watch the videos and try to draft in the form of a lecture notes. Okay, that will be definitely helpful for you. Okay, so we have completed biological databases in the first four sessions, okay, and then we have gone to the sequence analysis. We see the basics of sequence analysis, like what do you mean by next generation sequencing, and what is sequence alignment, what are the types, and even in next generation sequencing, do you know the types of next generation sequencing? So I'm not going to take something like an equipment based, but something related to the basic information, types of NGS, and then go for sequence alignment, types of sequence alignment like pairwise alignment, multiple sequence alignment. There is also another classification, global and local. What are these? We'll be discussing that. And we discuss about the amino acid substitution scoring matrices. And to the next class on Monday, most probably, I think I just ordered a digital pad. So if digital pad comes, then I can take this on Monday, or else I take some related to this fast and blast tools. And on Tuesday, I'll be taking this through the digital pad. Okay. So this is a problem based. Okay, I'll be teaching you how the algorithm works. A needleman versus waterman. Okay, either Monday or Tuesday, can postpone accordingly. Yeah, then we see even how the faster tools, the other tools of blast works. Okay, so this will be the end of the sequence analysis. So today we also see how the how to work with multiple sequence alignment and how the pairwise sequence alignment works. Okay, yeah. So let me take you to the today's session on I just share you the PPT. So if you see the sequencing, okay, so the well-known sequencing analysis of we have two different methods. Okay, the chemical method. So in 1970s, we generally go for Maxim and Gilbert method. I think this information is basic. Okay, basic information. The theory information I try to speed up, and if it is a practical hands-on, I can halt and I can show you demo. Okay, is it okay? Okay, I assume okay. So even you can search the information from the Wikipedia. You have plenty of information. Also find in textbooks. So what are the well-known sequence analysis? Sequence here we mean the DNA sequencing, defaultly DNA sequencing, and of course the secondary we take even how about the protein sequencing? If you see the sequencing, okay, generally we go for Maxim and Gilbert method. That's what we call it as a chemical method. Here we use four different chemicals to identify A, C, G, T. If you want to identify the nucleic acids in DNA, we use four different chemicals to identify ACGT paths. Okay, and similarly, enzymatic. Here we use polymerases. So in Sanger enzymatic method, that is from 1980 onwards. From 1980 onwards, I mean up to present, we are using the enzymatic method. Most of the NGS techniques go for utilizing the enzymes, and of course, it follows the modified Sanger method. Okay, and if you see the DNA sequencing, okay, the especially Maxim Gilbert sequencing, I told you we have four different chemicals to identify ACGT. But the idea is not like that to just separate ACGT. You can able to cleave A plus G sides, and you can able to cleave G and C separately, and C plus D. How this can be done? How this can be done? How the bands are formed, and how we can identify A from G? Because in this, if you use a chemical, A and G will be cleaved. Here, the second chemical will cleave wherever it finds a G, and third chemical cleave wherever it finds the C. And this is for both C and D. Then, what you can infer, you have to check the where exactly. So for this, I first tell you the chemicals, and I tell you how this idea is developed. First, if you see the purines, okay, in purines, adenine or guanine or depurinated using formic acid. If you find A or G during the sequencing, okay, then the formic acid is going to cleave the sequence. For example, if you see a sequence here, which is following from 5 prime to 3 prime, where you are using a kind of radionucleotide phosphorus is attached in the 5 prime end. Okay, so what is going to happen if you go for sequencing? Okay, like if you place a cursor here in computer, so if you place your cursor in the next space, then obviously it moves during the sequencing, the character your sequence is going to study base by base. So first initially it starts from G and goes to the C and goes to the T like that. Okay, then only it can able to find if it finds the G initially. Okay, you find a G here, but actually if you find a G, okay, after G you can find C. Okay, so if C is found after the G, immediately on the first base you won't get. So in the second base, when you go for a sequencing, you find a C. So just because you find a C, obviously get only G base. And here, if you see, you also find one C here. So before C, this sequence is going to be clear. Like you generally have a pool of DNA like this. For example, if you have this small piece of DNA, a pool of DNA, I mean some millions of DNA if you have. Okay, if that you can give as an input. So during the sequencing, so if it finds one base, okay, the sequencing is done by base. So it checks what base is there. According to that, you can get the fragments. You get some thousands of fragments like this, and here also you get some thousands or 10,000s. Here also you get thousands to 10,000. So if you have millions of DNA strands, whatever cleaving you have that much populations. Okay, it depends on the sequence. So if you find purines like A and G, these are purinated using formic acid. Okay, if you go to the next page, if you find A or G, the formic acid cleaves the strand. And the guanines are methylated by dimethyl sulfate. So A plus G, formic acid, and guanines dimethyl sulfate. If you can able to find, if you see the A plus G and G, see here you find an A. So you first initial GCT, and now you also see this G. How you find? One day you find A, isn't it? So you don't find GCT. Whenever it finds A, then you can, whenever you find G, then only it gets cleaved, isn't it? I can show you. Movement. So initially, if you see here, okay, so just because you find A, you find GCT. And if you see this G, G is find only after this. Okay, so you get a stand like this. GCT AC. Only one G is there, so you have only one kind of a stand. Okay, so but here you can find even if it finds G, so whatever stand you find here, you can find the same stand. Okay, and it also finds whether you have A also here. So obviously you have this kind of a stand. Okay, so likewise it can distinguish A plus G and G. Okay. And A plus G is depurinated by formic acid, whereas G is methylated by dimethyl sulfate. This is the second chemical. And the pyrimidine C plus T are hydrolyzed using hydrogen. Okay. And if you want to separate only the C alone, are going to add additionally a salt to the hydrogen. If you add this is the fourth chemical. Okay, first chemical formic, second dimethyl, the third hydrogen for the pyrimidines, and if you add an additional salt, only the C alone will be getting separated. So the modified DNAs may then be cleaved by hot piperidine at the position of the modified base. So this is how chemicals are used for sequencing, because if this is just a small sequence, but the DNA sequence will be not less than 10,000 bases. Definitely, it won't be less than 10,000 bases. Some will be 1 lakh, some will be 10 lakhs, and more than 10 lakhs also you can find bases in a DNA. If so, it's not easy to get a sequence through gel electrophoresis. You know, the gel electrophoresis maximum it has 24 wells, isn't it? 24 or 32, I don't know. So here we have 24, but how about in abroad? We may have, in US, we may have a large number of wells. So, but we can't have more than 50. Huh? So, obviously, one can cleave as much as small strands, and these strands are used for sequencing. This is a chemical method earlier used in 1970s, but in 1980s we got a good method developed by Sanger through enzyme. The enzyme name is DNA polymerase, which is used for separating, which is used for sequence identifying the ACGT. Okay, so here we use primer and DNA template, DNA polymerase, TDNTPs with fluorochromes, and DNTPs. Okay, so these are the reagents we use for sequencing. And if you see the sequencing by hybridization, here in sequencing by hybridization, this is the concept followed by Illumina sequencers. Okay, so it uses even a DNA can be hybridized to DNA, a single stranded DNA can be hybridized to DNA, or a single strand DNA can be hybridized to RNA. So hybridization is a kind of merging two different strands, can be the single strand of DNA or the single strand of DNA and obviously single strand of RNA. So this hybridization has happened through hybridization chamber. Specific conditions are maintained, temperature, humidity, etc. for hybridization protocol. And a kind of NGS, this is the second NGS kind of a technique. Okay, NGS, next generation sequencing, is a combination of so many sequencing technologies. So one I explained you like a sequencing by hybridization. The second is the nanopore sequencing. In nanopore sequencing, okay, you find the flow of electrons can determine the base. For example, if you have A here, okay, and in top you have G, and in top you have C, okay, each and every bases A, G, C, T can be identified based on the charge. Okay, if the electrons hit, okay, you know that ACGT is having a different chemical structure. Obviously, molecular weight is different, and even the conductivity. Okay, so that is also a different in these spaces. Okay, so you pass an electric current. Okay, a flow of electrons. While if you and in the vertical direction, no, in the horizontal direction, you pass electric current in the horizontal direction, and you pass the DNA sequence, okay, in the vertical direction. So each and every base will be passing like this, just like how you pass the cell in the flow cytometer. Okay, like this, a DNA sequence is going to pass, and obviously the electrons hit, and through the electric current, you determine what where you find A, C, G, and T. Okay, so the theory behind nanopore sequencing is that while when a nanopore is immersed in a conducting fluid, a potential voltage is applied, and so an electric current due to conduction of ions through the nanopore can be observed. Here they mean each and every base like ACGT has a different conduction. So through that only we identify that. And sequencing by synthesis. Here in synthesis we use fluorophores. Okay, so A, C, G, T are identified through four different color fluorophores. One is orange. If you think like, if A is orange and C is blue, red is G, and green is T, then this is how, if you just label it through the fluorophores, there's four different fluorophores. Only one base can take one color fluorophore. Now, obviously, the sequencing will be easier for us. Pulsed multi-line excitation. Okay, here you use four different lasers. Four different, we can also see the wavelength of the lasers, the blue laser, yellow laser, and something like a purple laser, red laser. Okay. So if you pass a sequence and if you have a four different color lasers, obviously these lasers can identify one is specific phase. For example, if yellow laser is for A and this is for C and this is for G and this is for T, only these phases are identified. Okay, in pulse multi-line excitation. So generally here you're not going to use a fluorophore, but you use a dye. You use four different color types: yellow color, blue color, uh, the purple color, and red color. Okay, these dyes can will be automatically identified by the four different color lasers. So you should also have these four lasers in your system. Then only it can identify these dyes. Okay, so this is pulse multi-line excitation. Microfluidic separation platforms, like how we find a capillary kind of a method during the nanopore sequencing. Here, we are not using such a thing, but here you're using a microcapillaries, okay, where you have used a kind of a chip, and through the chip, you can able to identify the sequencing. Okay, so this is a microfluidic separation platforms. So nearly 384 channels you can find in a chip, and so you can able to sequence up to 560 bases. Unlike gel electrophoresis, limit up to 24 bases. Here you can able to find nearly 560 bases can be found in a small chip. Shotgun sequencing. Of course, this is an embedded technique. Okay, one should generally apply the shotgun sequencing before going for the sequencing. So only you get uh, short strands. If only if you have short strands, it will be easier for us to deploy it in any technique. Okay, so most of the techniques, NGS techniques, also use an additional technique like shotgun sequencing to break into short strands. Okay, and now let me take you to the uh, sequence alignment. So, what are the types of sequence alignment? So, now till now, whatever we have seen is about the sequence analysis, types of sequence analysis like chemical and enzyme, metic method, and what are the types of NGS technologies. Okay, and yeah, I think the more about the NGS you can see in any other videos also. We have like Illumina, we have we have different equipments. So generally, like for if you want to go for a chromosome kind of a sequencing, or you want to go for a whole genome sequencing. Okay, so based on what exactly you want to sequence, we select the sequences based on it. Okay, you have one thing you have to remember that for going for sequencing, you are going to use a sequencer. Okay, and so whatever sequence you get, okay, from the sequencers, we go for pattern matching. Like, for example, if you are doing an experiment in your lab, I give you a sample like a Bengal gram. Okay, I dissolve in a kind of a solvent in a buffer, and I gave you this as a protein sample, a plant protein sample. And if I ask you to check what protein it is, what you do? Immediately you have to take it to the sequencers. Okay, you have to go to the sequencers. You have to use the preliminary steps, whatever we have to make it for the sequencer, and then you go for the sequencing ultimately. So whatever sequence you get from the sequencer, okay, you have to take it and you have to blast it. You have to check the pairwise sequence alignment. You have to check the pattern matching or the pairwise sequence alignment with the tools and databases available. Okay. So if you check like that, okay, I told you in NCBI protein, you have nearly 300 million protein sequences are there, more than 300 million protein sequences are there. If so, if you do a protein sequencing from the sample, whatever I have given, so you will be searching with the 300 million protein sequences. and you can able to find what is the exact protein name, okay, and what is the exact what is the exact plant species name. Okay, so if I give you a protein of from a Bengal gram, you can able to find only through the initially through the sequencer, but sequencer will just give you the sequence. It won't tell it is a Bengal gram particular legume hemoglobin protein something like that. So if you want to find even what is the exact protein, like even like hemoglobin, you have chain A, chain B, and you also have subtypes. Okay, so what is the exact protein that you can able to find only if you go for pattern matching, okay, or sequence alignment using the tools and databases available. Okay. So for going such a kind of a sequence alignment, okay, we have majorly two types of sequence alignment. That is, one is pairwise and the other is multiple. So whatever I mean sequence alignment, this is for both DNA and protein sequences, all types of protein and all types of DNA sequences. Okay, and if you see this sequence alignment, and especially in pairwise we can also have subtypes like global and local. Okay, so either you have to go for global or local. Most of the tools uses this kind of option. Okay, if you want to check it, if you want to check the local, it takes the local alignment, but it consumes more time and it's more precise. Okay, but if you want to complete it as soon as possible, then you can go for the global alignment. I tell you what the next slide about what is global and local. So before that, we can also go for the classification of pairwise sequence alignment. We have three classes majorly. One is dot matrix methods. We use tools like dot plot. We use tools like dot plot, dot let, etc. Okay, so for each and every method, try to practice with the tools dot plot, dot let. So we also have a package called EMBOS, European Molecular Biology Open Software Suite. So there you can find this program. And the second method is the word or K-tuple methods. Generally, FASTA, there is a tool called FASTA. You already heard about this term, isn't it? FASTA. What is FASTA? It is a format. It is a common format for the sequence. But this FASTA is different. That is, initially whatever I explained you from the first class is a format for a DNA or protein sequence. But this FASTA is a tool, is a pairwise sequence tool. And the expansion of this FASTA is fast alignment. Okay, only for this you can find this expansion, fast alignment. Okay, so this tool is going to use word or K-tuple method. Okay, of pairwise sequence alignment. Next, the third method is the dynamic programming. In dynamic programming, we have these two classes actually, local and global. Okay, in dynamic programming, if you want to practice with global, it defaultly uses the Needleman once algorithm. So more about the algorithms like Needleman once of global, Smith Waterman of local. We'll be discussing it on the next class. Okay, so there's a problem like how this algorithm works. One should also understand the algorithms. Bioinformatics is not just practicing the tools and databases. One should also know how the algorithms has to be developed and how it's working. Okay, it's working virtually, obviously, if you request and if you put any input any sequence, but you should also know how the output is coming from the input sequence from the tools. Okay, okay. Now let me go to the next classification, major classification of sequence alignment. That is multiple sequence alignment. Obviously, in multiple sequence alignment, you have to compare sequences like pairwise sequences alignment at a time. Okay, so I'll be explaining you. That is, here you are comparing more than two sequences. If you compare more than two sequences at a time, then we call it as multiple sequences. Here, you compare only two sequences. Okay, you have a sequence and B sequence. If A sequence is what you sequence from the sequencer and B sequence is from the database, then one can able to know uh, what exactly, whether is it exactly uh, a sequence, or if it is something unique, you can able to find some unique one. Okay, so you have to compare your unknown sequence with the database sequence. And so only the pairwise combination is uh, that is only two sequences comparison is done in pairwise. And if you want to link more than two species in the form of relationship, in the form of phylogenetic tree relationship, okay, then we go better go for multiple sequence alignment. For example, if you're working on heat shock protein, okay, myself, I'm working on heat shock proteins in cancer. If so, I'm initially working with in computational studies and trying with in vitro and followed by in vivo. So if I'm working on in vitro, so I'm going to use the human cancer cell lines. Okay, but we don't know whether whatever drug effect we get for the human cancer cell lines, the same effect we'll be getting in animal studies like mice or rats. So mice is now compared as one species, like a heat shock protein of human is once first sequence, and a heat shock protein of mice, if I try, that will be the second sequence. And if I try with the rats, okay, then that will be the C sequence. Then you should compare, okay, even how the sequences of a particular target looks like if you try to work on in vivo studies. Then only we can have an idea on what exactly and what is the mechanism behind and why the drug is working with one species and not working with other species. All this information can get given by multiple sequence alignment. So generally get a kind of evolution relationship using the multiple sequence alignment. And the methods are consensus methods, dynamic programming, genetic algorithms, hidden Markov models, iterative methods, motive finding, progressive methods, and simulated analysis. Okay, so these are the methods of multiple sequence alignment. So let us focus on to practice tool like Clustal. Okay, how the Clustal works. In Clustal, you have types of softwares like Clustal W and Clustal X. Nowadays we're using Clustal Omega, which is coming under Clustal W. Okay, so we see how this software works. Like the Clustal, we also have Tea Coffee, Pile Up. Okay, so many softwares are there for working for multiple sequence. And each and every software works on a specific methods, these specific methods. And if you see this kind of programs, okay, so the Multilink, so the and Cluster W, these are working on heuristic mode of alignment. Okay, so more about this we'll be discussing while working on that roads. Now let us discuss about local versus global. If you are comparing sequences as such, okay, if you are comparing sequences, I think you have a sequence like this and like this. So if you compare as such, we call it as a global. But if you want to get maximum number of matches, so related to the maximum number of matches, one should know what are the elements present in sequences. Then I can explain you about global and local, you can understand. For example, if I take you this kind of a sequence, so this is a large sequence. I can give you a small example. Think like you have A, C, T, A, C, T. Okay. And I have a second sequence like A, T. Okay. So think this is our sequence one and this is sequence two. And generally for these sequences, okay, you have three important technical terms. One is matches. If you find A and you compare with A, this we call it as a match. And in the second character, you can find here you find C and here you find T. This is mismatch. But here you can see the T is there in sequence one. Okay, if this is match, this is mismatch. Okay, match and mismatch you can find. But in the third character here you can find as T. But in second character, I don't find anything. That doesn't mean it is a gap. So if you don't have anything, you can't tell this is a gap. Because the gap, which is a third technology term, okay, technology term, which is generally found in between the sequences. For example, if you have a fourth character like T, okay, if you have a fourth character like T, and if you have something like G, okay, and if you have a cap like this, the cap is generally represented like this. So if just because the length of the second sequence is 4, but you can find in between a character is deleted in mutation. So how this you can find a cap? Mutation you find addition, deletion, substitution mutations, isn't it? If any deletion mutation has happened, you find a kind of a cap. Okay. So if it is in find in between the sequence, then we can consider this as a cap. Now the third one is a cap. Okay. And the fourth one is a mismatch. So one should remember during the sequence alignment, you find these three technical terms. Number one is match, number two is mismatch, number three is cap. Okay, these three are followed in each and every character comparison in pairwise sequence alignment. So here, if you have this as an example, okay, and if I give, if you find match and match, okay, for a match, if I give a score like +2, wherever you find match, you have to include as +2. And wherever you find mismatch, if I give you a score like -1, okay, so -1. Think if you find a mismatch, you have to give -1. And if you have a cap, okay, again, if you have to give -1. Okay. So these scores, whatever I mentioned, matches two, mismatches -1, cap as -1, because we anticipate only matches. So we are checking which is more close to our database sequence, isn't it? If this is your unknown sequence, this is what you are going to try. You are going to check with the database sequence, okay, and you have to identify. You have to come with the solution like what sequence, what protein, and what species it is, isn't it? If I give this kind of a scores, okay, and these scores are generally used for calculation of alignment score. The alignment score formula is how many number of matches into correction factor, whatever I given to, and number of mismatches into correction factor, that is -1, and number of gaps into correction factor -1. So if alignment score is a combination of matches, mismatches, and gaps, okay, with respect to correction factor, if you calculate the alignment score here, okay, think like you're calculating alignment score. So if you compare sequences as such, this comes under global alignment. In global alignment, okay, you find one match and two mismatches, isn't it? So obviously, what is the calculation? 2, okay, -2. And how about gap? One gap. So -1. So the alignment score in our case is -1 for global alignment. If it is local alignment, we can adjust sequence in such a way that we can get maximum score. Our objective is to get maximum alignment score. Okay. So what I'm going to do here is, if you have a sequence, sorry, I'm checking. I'm writing once again. A, C, T, T. Okay, think this is your first sequence. And now I'm going to write the second sequence. A. So I'm going to give a kind of a gap here. Okay. I'm going to give a gap here. Hyphen T, hyphen G. You got it? A C T T, A gap T hyphen G. We can check the score now. Here you find one match. Here you find another match. So two matches you can able to find. How about mismatches? So C and hyphen. Okay. This is a gap. This is also a gap. If this is a gap, this is also a gap. But here you don't find anything, isn't it? So this is a mismatch. This alone is a mismatch. This is a gap. This is a gap. Okay, but these two are matches. So you have -3 here. you have two gaps and mismatch. According to the correction factor, okay, totally you have minus 3 here, and you have two matches. If you have two matches, 2 into 2, because the correction factor is 2, isn't it? So the alignment score in our case is for matches, you find two matches and you are multiplying by the score of match. So you have 4. And similarly, you gaps and mismatches because they are given the same score. Okay, you just sum up, you got like 3, and 3 into the correction factor is minus 3. So in our case, now we got alignment score as plus 1. Okay, you understand now? So in local alignment, we majorly focus on adjusting in the sequence in such a way that to get a maximum score. Earlier we have minus 1, now we got like plus 1. This is what we call it as local alignment. Okay, one should also think about to go for a local alignment to get a kind of a precise sequence. Okay, so the alignment score, whatever I mentioned, okay, this extract is taken from a textbook called Bioinformatics Computing by Bergeron. Bioinformatics Computing by Bergeron. Okay, so this example they have solved. You can try the same. Okay, so however I mentioned, they have calculated that. Now, let me explain about the another problem. This is related to edit distance. There are two types of edit distance. One is Hamming distance, the other is the Levenstein distance. If you find similar characters, okay, like for example, if this is sequence 1 and this is sequence 2, okay, just because the characters are same, okay, if only if the two strings are of equal length, then distance is applicable. Okay, then what is distance here? The distance, whatever I mean, is the genetic distance. So genetic distance is nothing but mismatches in the sequence. Think like this is if, a father sequence and this is a son sequence, okay, of a particular protein. Okay, I mean, if you see a particular DNA of that father and son, okay, you can find two mismatches, isn't it? G and T are matches. G, T are matches. But you find two different mismatches. Whatever mismatches you find, not only mismatches, even if you find gaps, that are all included as genetic distance. And obviously, that genetic distance is what? Hamming distance. And similarly, there is another distance called Levenstein distance. If you have unequal characters in length, then you can go for Levenstein. It doesn't mean that for equal characters, Levenstein is not applied. Levenstein distance is also applied for equal distances. But if it is an unequal characters, for example, for the first sequence you have AGTC, but also you have think like you have another character like C. Okay, but whereas in second sequence you have only CGTA, then Hamming distance is not applicable. You have to go for Levenstein. If you go for Levenstein, again you find two mismatches, and also this is considered as a mismatch because here even if you find a gap at the end, you won't be considered as a gap, but you have to consider this as a mismatch. So can be mismatch or gap. Obviously, you have to sum up. So what you get in this case is now 3. Okay. So but you can't go for having, but whatever you calculate is the Levenstein distance. Okay, so these are some of the concepts. Okay, and then I can go for the pairwise sequence alignment tools. So in this URL, molbio-tools.ca, you find so many kind of sequence alignment tools, especially for pairwise sequence alignment. Okay, molbio-tools.california, isn't it? No, Canada. Okay, you can find this much tools. And methods of PSA. Now we discuss what is dot matrix method. So in dot matrix method, okay, you are going to have initially sequence 1 in the rows, in the columns, and sequence 2 in the form of rows. The sequence 2 is filled in the row wise. So if you were comparing sequences, characters in the sequences, okay, you deploy a matrix. Okay, you go deploy a two-dimensional matrix and you try to input all this information. Okay, so what is the information you have to fill it up? If you fill the sequence in the column wise and the sequence 2 in the row wise. You have to fill wherever you find matches, you have to fill asterisk. So think, see the first row alone. You find in the first row you find A. Okay, see the columns here. You don't find any column A. Only for this alone you have A. So you have to put an asterisk. Now go to the second G. In G, you have to check for the G. Yes, you have a G, you put a star. Here also have a G, put a star. Here also have a G, put a star. Okay, this is how you fill the dots. Okay, from the top to the bottom. Whatever you filled, okay, generally, if you are going for pattern matching, generally, when you go for pattern matching or sequence alignment, you'll be checking like to identify the sequence, isn't it? So most of the sequences you find the matching characters are present in the tag. When compared to this, you have only two continuation. But see here, you have four continuation. But you have mismatch. But again, you find two continuation, three continuation. This can't be find here, somewhere here, somewhere here. Okay, so mostly you find matches in the tag. So that's what they have shown here. And this output is taken from the emboss dot matcher. So there is a software, there is a package called emboss, European Molecular Biology Open Software Suite. Here you have 151 programs for pairwise alignments, multiple sequence alignment, and other kind of sequence related problems. Okay, so we can able to work it out with emboss. Okay, if you see dot matcher is the program. Like dot matcher, I already told you. Dot plot, dot let. These are other programs you can use for getting this dot matrix table. So getting this dot matrix table is what we call it as a dot matrix method. Okay, that is after putting dots. So you have to check for the optimum thing, which is present obviously in the diagonal. They just show this, join the diagonals and show this as a dot matrix method. And now go to the second method, word or k tuple method. This is followed. This is followed by the FASTA program. FASTA, I already told you, fast algorithm. Okay, so this is the extract taken from a textbook called Bergeron, Bioinformatics Computing by Bergeron. So in the textbook, okay, here you can able to find uh, a hash table, a query sequence. Okay, hash table and query sequence. The like generally we have to start from here. You have a query sequence like sequence one and database sequence like sequence two. Okay, one, two. Okay, so think like you have a sequence one and sequence two. The query sequence, you're checking the pattern matching with the database sequence. So our first codon is AAA. Okay, so if you have a codon like AAA, okay, and generally you know the codons code for amino acids, isn't it? So if you see the amino acid single letter code and obviously the codons, okay, you obviously have 64, but only 64 code for amino acids. So only 61. The remaining three or stop codons, you won't code for amino acids. So in this case, okay, whatever you find in the hash table, okay, so initially if you see the initial codon A, okay, this is present in first position, isn't it, in our query? You have to search in the database. The same AAA, you have to check the position. So you see here, 1, 2, 3, 4, 5, 6, 7, 8, isn't it? In eighth position, you can find the same AAA. So in query, it is in first. Okay. In the and if you see in the database, it's in the eighth position. And now you have to fill something instead of filling asterisk like in dot matrix method. You have to fill only the numerical value. How you got this numerical value? For this, you are going for the index table, hash index table. So in the hash index table, like for the 64, they have mentioned the possibility of a combination of three characters, that is nucleic acid bases. So if you see here, In the index one, it find the same AAA. So what do you understand from that? This value one has to be filled here. The matching of query and database. So here it's in first position, and here it's in the eighth position, but here it's in this is what one have to be filled now. This I filled here. Now see the second query sequence ACG, where you can find the ACG. ACG is present in seventh position. So see the in seventh position we are filling, but what we filled 12. Where we got this 12? We got from the table. So what we do, we are hashing through the key and values. This is what we find a concept in Perl programming. Okay, so hashing, that is like a pointer and address in C programming. Okay, in Perl we call that as a key and values. Okay, the same concept they use like that. So which we can able to find in this FASTA algorithm, fast algorithm. So the this kind of algorithm is shown in this flowchart. Okay, dynamic programming. In dynamic programming, I told you there are two types. One is Neeleman and Smith-Waterman. Okay, and if you see about the Neeleman Smith-Waterman, I'll be explaining you on the tomorrow's, I mean, next session class. In multiple sequence alignment, where in the same URL you can able to find. Okay, like so if you drag down, you can find the multiple sequence alignment tools. Okay, cluster W, cluster omega, MAFT, LLine. So in the same website, EBA website, you can find these tools. So we can also find in the second page, you can also find some more tools. Okay, multiple tools are there. Nearly some 20 tools are shown in the single website. Okay, if you click that, it takes you to the specific website where you can practice that. Okay, the major multiple sequence alignment, what we see initially is a progressive strategy. Okay, in progressive strategy, you can find pile up, cluster W, cluster X, MSA,
नॉइस
program
हो
block.
रहा है क्या?
Uh, in progressive strategy, yeah, the participant can, if you have a doubts, you can ask me or you can find it. Yeah, I found some question from Jyoti Rawat. Sir, we should always flash context sequence obtained after sequencing. What in case if we get forward and reverse sequences separately? How will we will make it context? Yeah, you are speaking related to sequence assembly. The sequence assembly is something different, okay, which we will be discussing it. Okay, related to the context, all these things will be discussing during this session. Okay, last before mini project session. So during that time, I can teach you because there are so many concepts in sequencing, isn't it? Like context, you have so many terms, terminologies. One don't understand what you mean by context if I try to explain. Okay, I will see in the last before mini project discussion session. But okay, now let me take you to the illustration of the tools. Okay, we have less time. Okay, so then you'll be missing something. Okay, so if you want to get a kind of a protein sequence, okay, what you do, you go for the uniprot, isn't it? In uniprot, you can take a sequences. For example, if I want to get want to compare insulin for three different species, if I want to compare insulin ligand, okay, of three different species, first I am selecting the Homo sapiens, and related to the diabetes experiments, even they are trying with the zebrafish as the model. So we can check how about the insulin protein is matching with the human. Okay, for that I selected zebrafish. What else we can do experiments in our lab? We can try with the Sususcrofa is what used, okay, initially before genetic engineering comes into play for the pro-insulin, fast insulin, etc. Okay, so before genetic engineering, so many people sacrifice the pig for getting the insulin, isn't it? So we can compare pig as one of the sample. And if you're trying such a kind of experiments with rat model, okay, so I use these four sequences. Okay, I selected these four sequences. Now I'm going to go for downloading this sequence in FASTA format. If I click download, you get like FASTA canonical. Click download. Okay, so I'm going to save it in desktop. In Bioinformatics demo, this is actually an insulin 4 sequences, isn't it? So insulin 4, I just typed like insulin 4. It's coming as a compressive archive. No, no, no. I don't want compressive. Again, I'm clicking download. Compressed, no. I want in the form of a FASTA format to download. Now I'm going to give like insulin 4. Okay, defaultly it will be in FASTA file. No doubt in that. But I'm going to save it in my location. Insulin 4 proteins dot FASTA. Yeah, we can check whether it's in FASTA format or not. Yes, it's a FASTA format file. FASTA file. Okay, now the next objective is to go for multiple sequence alignment. For this, I'm going to take cluster W or cluster omega. So in cluster omega is present in EMBL EBA. If you want to practice with cluster W, which is an online tool for multiple sequence alignment, where progressive, this tool is works on progressive alignment. Okay. So for this, if you want to work on iterative alignment, you can go for hidden Markov models or neural networks based MSA. Now I just open the cluster omega of EBA. Meanwhile, I can open another server that is DDBJ. In DDBJ also we can find cluster W. Okay, so I type DDBJ cluster W because whatever output you can find in EBA, you can't find in DDBJ, and whatever output you can find in uh, EBA, I mean DDBJ, you can't find in EBA. So I try both the websites now. So I just open the cluster W of DDBJ initially. So initially I have to input a sequence. Okay. So I'm checking my kind of input where I can input. This is right to work DDBJ cluster W. No, at gateway. Some problem is there. So I can check with the initially with the EBA now. So I have to input either I have to copy and paste the sequence. Okay. Like if you just open it in Notepad, it will be better for you. You can also use Mega software. Okay. Either copy and paste or else you already saved it, isn't it? So just upload it. Okay. You're not copying and pasting, just upload this. So for this, you choose the file from the location where you have saved in desktop, bioinformatics demo, go to the incident 4, click open. Okay, you choose the character default. You can give some name if you want. Yeah, we got an output like this. So this is how the four sequences are compared. Okay, and related to the alignments, you can get some information. Okay, kite tree. So which is more closely related to with each other. You can find human is closely linked with the pig, according to this. Did you find this? So human is closely linked with the pig. Okay, that's what you can find in the tree relation. And when compared to human and pig, rat is related. Okay, so the next species, zebrafish, is distantly related. This is what you can find in the phylogenetic tree relationship. Okay, this is from EBA. I'm checking any possibility to work with TTPJ cluster W. Similarly, can try with even genome net cluster W. The cluster W is software present in uh, various public domain databases. From the databases itself, they embed these kind of a tools, like how you find blast in most of the databases. Even cluster W is present, but in few databases you can find. Um, genome net. I can check this. Yeah, it's asking for a format. Choose. I'm going to give insulin 4. Execute multiple alignment. Even in Embos, you can find. Okay, this gives a kind of a best comparison. Okay, in genome.jp. Okay, if not DDBJ, which is of course from a Japan. This is also a product of a Japan, that is genome.jp. So in genome.jp, in cluster W, I just showing you an output. So the first sequence is zebrafish. Okay, the zebrafish score is very less. If you find more score, then it is more best. That is the best alignment. If you see here, this is the best alignment. 2 and 3. Human and Picor, the 110 amino acid 1A08 is having more matches. If and if you have more matches, you get more alignment score. This is how you can able to create the sequence alignment. So whatever you have seen for the multiple sequence alignment, for pairwise sequence alignment, we see the illustration on tomorrow. I mean the next session. If you have any doubts, you can ask me. I have completed my session on today. Do you have any doubts? Okay. Thank you for the interaction from the beginning. Okay. And if you, even if you have any doubt,